All right, so today I'm going to be talking about some of the current research from our lab, and specifically on antimicrobial Th17 CTLs targeting this bacteria that we refer to as Acne. All right, so we all know that Acne vulgaris, so this is a disease of the pilosebaceous unit, and a number of factors have been implicated in its pathogenesis. So today I'm mainly going to focus on how different strains of Acne drive the Th17 response, which can either be inflammatory, and a recent work from our lab, we've identified some subsets of these Th17 cells that can actually have a protective effect. So Th17 cells, just a brief introduction. So these are subsets of CD4 T cells, and we know that in the periphery, naive T cells, upon encounter with APCs, they normally differentiate into different subsets under the influence of different cytokines. So these cells, Th17s, they've been implicated in providing immunity to extracellular bacterial, fungal infections, and we also know them to be very potent inducers of tissue inflammation. So they express the transcription factor is retinoic orphan receptor C, and upon activation, they produce different molecules, including IL-21, IL-22, IL-17, CCL20, and IL-26. And the signature cytokine is IL-17, and this cytokine can act on both immune and non-immune cells, leading to the recruitment, activation, and migration of these cells to sites of inflammation. So some data from our lab, if we sample the pilosebaceous unit of acne patients, and from normal skin, what we see is that we see elevated IL-17 levels from acne lesions as compared to acne skin. And if we do immunohistochemistry, again, this is on acne lesions. We see that we can see IL-17 expressing lymphoid cells around the hair follicle and around the comedone, suggesting again that IL-17 is implicated in the pathogenesis of this disease. And then recent work from Fitzgibbon, the Lee group at UCLA, they also did metagenomic analysis of Acne strain populations, and what they found out was that they observed 5 major microbiome types, and so sampling the acne patients, they found that ribotypes 4 and 5 are mainly expressed in acne patients, whereas in the normal patients, they could only see the ribotype 6. So in our studies, what we did is we selected 6 representative ribotypes, so the ones that are associated with acne disease. So I'm going to be sharing with you data whereby we use ribotype 4 and 5, and then for the healthy associated strain, we use these 3 strains are mainly of ribotype 6, and you're going to be hearing me talking about PA, and these are strains that are associated with acne disease, and PH, these are strains that are associated with healthy skin. So our hypothesis, so we hypothesize that Acne ribotypes trigger Th17 cells that either contribute to pathogenesis of acne, or they can lead to a protective mechanism. And the question we wanted to address is, could the immune response to these P and PH ribotypes be different? So the first thing we did was stimulate peripheral blood mononuclear cells from healthy patients or normal donors with the acne-associated and the healthy-associated strain. And what we saw is that the acne-associated strain induced significant levels of IL-17, and this suggested that these strains have an ability to modulate the immune response at the T cell level. So the smart thing to do, we decided to clone Th17 clones. So basically what we're doing is we are taking Acne and the healthy-associated strain, stimulating PBMCs, and then we're selecting for a double population of cells expressing CD4, secreting L17. We do sterile cell sorting, and then we expand these clones in culture. And so our cloning efficiency, so we're able to get clones, our cloning efficiencies between 26 to 32%, and we test for the specificity of these clones in T-cell proliferation assays. So these are some of the clones. This is an acne-associated clone. Again, you see that these clones they're specific for the molecules we're interested in, so they are secreting L17. These are the healthy-associated strains. So both of them are secreting L17, and when we do real-time PCR, we see that they express molecules that are associated with the Th17 phenotype. So the next thing we did was to try and look at the cytokine profiles of these clones. So the healthy-associated strains, they induce what we've renamed protective Th17s. I'm also going to be calling them Th17 CTLs, and I'm going to share with you the data in a moment. What we saw was that despite producing L17, L22, and IL-26, these clones also were secreting IL-10, and we know that IL-10 is a modulatory cytokine. And what we decided to do next, we wanted to see if this, if we activate these clones, then we get the supernatants. Can these supernatants be able to kill P. acne in vitro CFU assays? And just to share with you the data here, when we do these CFU assays, we see that supernatants derived from these protective Th17 cells are able to kill P. acne. And you can see more than 5-log reduction in the CFUs. And then if we do electron microscopy, we see that the mechanism of killing is through pore formation of the bacterium membrane, leading to cytolysis. The molecule that's responsible for the killing is heat sensitive, because if we heat inactivate these supernatants and then do the CFU acid, we'll lose the killing ability, suggesting that this molecule may be a protein. All right, so for the acne-associated strains, we saw a totally different picture. Upon activation, these clones, apart from secreting the Th17 signature cytokines, they're also producing interferon gamma, which is a Th17 cytokine, and it's known to have an inflammatory response. So we've labeled these clones as pathogenic Th17s, and the reason for this is because if we do the functional assays, again, the CFU assays using these supernatants from these activated clones, we don't see any killing ability. So these clones are not able to kill P. acne, and you can see the picture here on the EM. All right, so this raises interesting questions, because there's a paper that that came out a couple of years ago, and they showed that Th17 cells, they promote microbial killing and innate immune sensing of DNA via interleukin 26. And we know that interleukin 26 is cationic, it's amphipathic, and normally it was shown to be able to kill both gram-positive and gram-negative bacteria through pore formation. So if I can go back here, all our clones are producing IL-26, so the question was, what is this IL-26 doing? So the smart experiment to do, we generated a clone that's secreting tons of, I'm going to use the word tons of IL-26 here, and also IL-17. We do the CFU assay again, we don't see any killing. So telling us there's something special about this P. acne cell wall. We use the recombinant human IL-26 again, we don't see killing, and then EM pictures, you know, the acne, they look happy, we see their normal pleomorphic structure. So we're still interested in identifying these molecules involved in killing. So what we decided to do, we took supernatants from these protective Th17s, incubated them with a neutralizing antibody 1226, trying to see if we can neutralize the killing effect, and we see that the neutralizing antibody has no effect. We tested recombinant human IL-26 against other gram-positive and gram-negative bacteria, E. coli here, and Staph aureus. And what you see is that recombinant IL-26 is able to kill both Staph aureus and E. coli. And if we neutralize the IL-26, again, we're able to inhibit this killing. So again, this suggested that IL-26 is not potent against P. acne, and there's something special about this bacterium. So here we are. Oh, sorry. So here we are. We're still interested in identifying which molecules secreted by these Th17 CTL cells and what is the mechanism. So I normally tell people in biology, if you can't figure out something, the best thing to do is just do bulk RNA-seq, right? So that's what we decided to do. So we got 20 clones, so 15 killer clones, and 5 non-killers as control. We stimulate this with anti-CD3 and CD28. We collect mRNA at 6 hour and 12 hour time points for RNA-seq. We use the supernatants from these clones for CFU assays and for ELISA assays. And then we can see if we can correlate the RNA-seq data with the ELISA and the CFU data. Just to share with you some of the data. So again, I encourage you to come and visit my poster. So my poster ID is 051, and we can talk about this in more detail. But the most interesting thing that we found out was these cytotoxic molecules that are normally thought to be expressed by CD8s and the NK cells are the ones that are highly expressed in the Th17 CTLs. Again, showing that Th17 cells can also be cytolytic. So we have granulysin, for example, here, the gene symbol for granulysin, granzyme A, granzyme B, and also we have perforin. So having seen this, I was kind of very excited, so I went back at the supernatants. I wanted to see if I do a quick ELISA experiment, if I can track down all these proteins. So again, I just summarize here. I picked 4 Th17 CTL clones, and what you see is that all these clones are able to secrete granzyme B, granulysin, and perforin, and these are the non-killers. So the non-killers don't express these cytotoxic molecules. If I do FAC staining, again, selecting clone 1731, again, they express very high concentrations of perforin, granzyme B, and granulysin. So this is very, very exciting. And so the next thing we've done is we started correlation studies correlating antimicrobial gene expression in RNA-seq with ELISA functional data. Excuse me. And what we see is that if you look at the normalized count of granulysin against the ELISA data, we see very, very good positive correlation, and you see the same with granzyme B and perforin. And then for the CFU assay data, we can also correlate the amount of killing with the amount of protein expression. So the more granulysin you express, the more, the more killer. So very good negative correlation. We are just starting to understand this. So we've come up with a model. So we think that this protective of the Th17 CTLs, they produce these cytotoxic molecules. So they produce granulysin. Granulysin acts on the membrane of the bacteria, gets inside. Once this pore is formed, then other molecules can be granzyme B, can be perforin, or other molecules that we are yet to discover. They get into the cell, they're able to disrupt protein synthesis and disrupt metabolic enzymes. On the other hand, for the pathogenic Th17s, they produce IL-26. This IL-26 is not able to penetrate the P. acne bacterial cell wall. We don't know why, so we are kind of starting to do studies to kind of elucidate that. And then the other things you're interested in studying is to see if these cells can trans-differentiate. If this cell, for example, can differentiate into being protective, or if the protectives can also differentiate into the pathogenic phenotype within the pilosebaceous unit. So again, it's very exciting, and I hope to share with you guys more data in the next ARS meeting next year. All right, so again, summary, we've identified new subsets of Th17. These are Th17 CTLs. They mainly secrete IL-10 they mainly secrete IL-10 in addition to Th17. Natural IL-26 is not potent against P. acne. And these protective Th17s, they produce granzyme B, perforin, granulysine, and other molecules that we believe are in there, but we are yet to discover them. And this is the last slide, so I'd like to give thanks to my mentor, Robert Maudlin, and my former postdoctoral mentor, Jenny Kim, all the collaborators. I'd like to thank the Acne Q Alliance, the AARS, and I'm currently being funded by the NIAMS. And again, thank you for your attention.
We have time for one question for Dr. Agak in the back. One question, thanks.
So have you tried to plot granulysine or the supernatants?
Have I tried to?
Have you tried to plot
Oh, that's the next set of experiments.
Yeah,
So
just to verify that that's the way they.
Yes, so I've done, I call it the classical experiment, so I've done one of those. The only problem I'm having is getting the granulysine neutralizing antibodies that work. I'm still kind of shopping around, but the one I have, when I did that, I see 1 log reduction in the bacterial CFU. So I think it's the antibody, but I'm going to do more experiments to see if it's the granulysine alone or if there are other molecules responsible for this killing. Again, thank you for the question. We are headed in the same direction.
Thank you, George. Thanks so much. Thank you.